Très heureux d'être ici avec vous cet après-midi pour souligner le lancement des conférences Télesphore-Parizeau. J'aimerais d'abord saluer tous les participants, les participantes. Vous êtes nombreux ici au M415 et aussi à distance. Et remercier tout particulièrement, souligner la présence de monsieur parmi nous, monsieur Robert Parizeau, car si nous sommes réunis ici aujourd'hui, c'est grâce à vous. Votre généreux don nous permet d'honorer la mémoire de votre grand-père, le docteur Télesphore-Parizeau, et d'instaurer les conférences qui vont être des conférences annuelles qui portent son nom et qui auront pour thématique les grands enjeux de santé de notre monde contemporain. Alors, pour cette première édition des conférences Télesphore-Parizeau, la Faculté de médecine a confié au département de microbiologie, infectiologie, immunologie le choix de l'invité. Nous aurons donc le plaisir d'accueillir le docteur Omar El-Halfawy, qui est professeur adjoint au département de biochimie de l'Université Régina, donc Welcome to Montreal, et qui présentera ses travaux sur la résistance aux antibiotiques. Mais pourquoi avoir choisi le département ou un thème lié à la microbiologie? Vous allez voir, c'est assez évident quand je vais parler des biographies, mais j'aimerais vous raconter les parcours inspirants du docteur Télesphore-Parizeau et de notre donateur, M. Robert Parizeau. Commençons par le donateur, M. Parizeau. Robert est un homme d'affaires accompli, un administrateur de société chevronné, expert en gouvernance d'entreprise et mécène des arts, diplômé de HEC Montréal. Il a consacré sa carrière au domaine de l'assurance et de la réassurance. Parmi ses nombreuses occupations, il a présidé le conseil d'administration de l'Institut de recherche clinique de Montréal, de même que celui du Fonds de solidarité FTQ. En 2016, il a été nommé officier de l'Ordre national du Québec et en 2004, il a reçu un doctorat honoris causa de notre université, l'Université de Montréal. M. Robert Parizeau fait également partie d'une famille bien connue du Québec et son grand-père, Télesphore-Parizeau, a été un pionnier en matière de bactériologie. À travers son riche parcours universitaire, on va redécouvrir certaines des étapes très importantes de l'histoire de notre faculté qui vient toujours de fêter ses 180 ans. Alors, après un passage à l'École de médecine et de chirurgie de Montréal qui a précédé la formation de notre faculté, Dr Parizeau a entrepris une formation médicale à Paris où il rencontre des scientifiques de réputation internationale. Premier stage en 1894 à l'Institut Pasteur de Paris où il s'initie à la théorie microbienne et aux techniques bactériologiques. Et de retour à Montréal comme pathologiste et chirurgien consultant à l'hôpital Notre-Dame, il offre des cours de bactériologie, de stérilisation et désinfection des plaies qui vont contribuer à mettre en place des nouvelles procédures opératoires en réduisant de façon significative les taux d'infection et de décès post-opératoires. 1896, il occupe un poste d'assistant en histologie et pathologie dans la nouvelle faculté de médecine et il est appelé à prononcer une conférence intitulée « Pasteur et le rôle des microbes dans les maladies ». Donc, 128 ans plus tard, les enjeux reliés à la microbiologie, aux maladies infectieuses sont encore et toujours d'actualité. En effet, la résistance aux antimicrobiens est l'une des 10 plus grandes menaces auxquelles l'humanité est confrontée si on sur l'Organisation mondiale de la santé dans un communiqué de 2023. Dr Parizeau a été nommé professeur titulaire en 1906. Il est délégué à Québec pour assister à la présentation du projet de loi qui donnera naissance à l'Université de Montréal en 1920. Il a donc participé très activement à la création officielle de notre faculté, mais aussi de notre université. Il a été nommé premier vice-doyen de 1922 à 1934, puis doyen de la faculté de 1934 à 1938. Il participa d'ailleurs activement à l'élaboration des plans du nouveau pavillon principal de l'université, le cher pavillon Roger Gaudry, dont l'architecte Ernest Cormier, comme vous le savez, a été l'architecte et où nous sommes présentement. Durant son décanat, le Dr Parizeau contribuera entre autres à la création de l'Institut de microbiologie et d'hygiène qui sera à l'origine de l'Institut Armand-Frappier. Il a été nommé chevalier de la Légion d'honneur en 1935 et obtiendra lui aussi un doctorat honoris causa de l'Université de Montréal en 1942. Donc, Dr Télesphore-Parizeau, beaucoup de réalisations, il fait partie des grands scientifiques et des bâtisseurs qui ont fait la réputation de notre faculté, de notre université. Et grâce au soutien de son petit-fils, M. Robert Parizeau, nous avons demandé à l'historien de la faculté, M. Denis Goulet, de publier sa biographie que vous pourrez vous procurer à la fin de la présentation pour ceux qui se joindront à nous au cocktail. Alors, notre mission d'excellence se poursuit encore aujourd'hui, notamment grâce à la création récente du Centre d'innovation biomédicale, qu'on appelle affectueusement le cible, qui est un projet phare de la faculté et qui regroupe les chercheurs et chercheuses des départements de sciences fondamentales sur le campus. La microbiologie est au premier plan de ce centre et nous avons pu recruter des chercheurs de renom tels que le professeur Yves Brun et la professeure Frédéric Leroux, respectivement détenteurs d'une chaire de recherche du Canada 150 et d'une chaire d'excellence en recherche du Canada. Nous avons eu effectivement effectué d'autres recrutements avec Sven Van Tiffelen et d'autres qui sont actuellement en cours. En terminant, je vous donne une petite primeur. Je suis très heureux de vous dire que nous allons bientôt annoncer un don de 1,5 million de dollars d'un donateur pour notre projet de recherche sur le Centre d'excellence sur les maladies infectieuses et préparation aux prochaines pandémies. C'est un projet de collaboration, de partenariat entre les chercheurs du cible au département de microbiologie et l'Institut Pasteur de Paris. Donc, encore une fois, après 130 années, après la formation du docteur Parizeau à Pasteur, nous continuons d'exceller dans le domaine de la microbiologie. Alors, avant de céder la parole au directeur du département de microbiologie, immunologie et infectiologie, le docteur Jacques Thibodeau, je me permets de vous présenter une petite vidéo du 180e anniversaire de notre faculté. Et sur ce, je vous souhaite une excellente conférence à tous.
Monsieur le doyen, Monsieur Parizeau, chers collègues, bienvenue. J'avais préparé un petit discours sur Monsieur Télesphore-Parizeau, mais je vois que Monsieur le doyen a bien fait ses devoirs et a repris l'essentiel, en fait, de la biographie du docteur Parizeau. So what I'm going to do, going to introduce our speaker for today. Dr. Omar El-Halfawy is coming from the Department of Chemistry and Biochemistry at the University of Régina in Saskatchewan. He's Assistant Professor of Biochemistry in the Faculty of Science. Since August of 2020, Omar holds a prestigious Tier 2 Canada Research Chair in Chimiogenomics and antimicrobial research. Dr. El-Halfawy has completed his studies in pharmacy at the University of Alexandria in Egypt in 2005. In 2009, he completed a master's degree in pharmaceutical microbiology at the same university. He then joined the University of Western Ontario to complete a PhD on the mechanisms of antibiotic resistance in the lab of Miguel Valvano. And from 2015 to 2020, he was a postdoctoral fellow at McMaster University in Hamilton under the supervision of Dr. Eric Brown. As a PI, he uses an interdisciplinary approach spanning microbiology, biochemistry, molecular and chemical biology, and chemogenomics to uncover novel antibiotic resistance and microbial virulence mechanisms and new antimicrobial solutions. So without further ado, please join me in welcoming Dr. Omar El-Halfawy to the University of Montreal.
Thank you so much for the introduction, and thank you all for coming, Mr. Parizeau. Thank you for the opportunity, and it's quite an honor to be here and in starting this presentation to you. And I'm really excited to share with you our work on the antimicrobial crisis. And it is quite fascinating, and reading the biography of Dr. Télesphore-Parizeau that at the time there was an infectious diseases crisis. There's a problem, and that was the pre-antibiotic era probably, and that's we're still living that crisis, and we're at the threat of having a post-antibiotic era. So if we look at the timeline here, there is certainly a decline in antibiotic discovery. Looking at the timeline for discovery of antibiotics that are actually introduced to the market, and you can see that over time, after the golden era of antibiotic discovery, that led to fewer and fewer new classes of antimicrobials being introduced until there was a discovery void. Now that's a little bit outdated. There have been some developments in the recent, like the last decade, but that's nowhere near what is needed to counter the problems that now here we see. In that, just again, just an example of the rise of antimicrobial resistance that leads to an increase numbers of fatalities worldwide. We have estimates of 5 million annually that are associated to antimicrobial resistance that can climb later on if the problem is unchecked. And so there are clearly a crisis in terms of infectious diseases, antimicrobial resistance. The antibiotics that we have are not adequate fighting that problem. But that's we're kind of losing at two fronts here. We're seeing therapeutic failures. Like I mentioned, there is the rise in antibiotic resistance, but also there is microbial virulence, that there are no therapeutics right now that are targeting virulence, which is the ability of microbes to cause those severe infections. And so that causes a problem in the clinic, but there are no therapeutics that are countering virulence. And then there are discovery roadblocks. So there are different reasons, and this is not an exhaustive list of why we don't have too many classes that as much as we need in terms of new antimicrobials. But there have been at some point, for example, so from the examples here, at some point there has been a shift in how antibiotics were discovered several decades ago, and that like in vitro target-based screens for antimicrobials, they didn't materialize that much success. And sometimes we have problems identifying the mode of action of new antimicrobials. So again, cannot be further developed later on to the clinic. And early on in the success in the golden era of antibiotics, a lot of them are natural product derived. And then later on, there was a problem of rediscovery. So we went through cycles of rediscovering the same antibiotics over and over again. And so that was also led to several setbacks. But also there has been consistently a focus on lethal antimicrobials or antimicrobials that either suppress or kill bacteria. So the traditional kind of approach towards kind of infectious diseases have an antibiotic that have to target an essential target in bacteria so that they suppress or inhibit their growth, which then also limited our kind of chemical landscape of what can possibly interact with those, what we think are essential targets. So clearly we need out-of-the-box solutions, we need different directions, and there's been emerging interest in antivirulence agents and also antibiotic adjuvants that kind of revive the pipeline and the antibiotics that we currently have. So kind of working on both sides, and I've worked on both antivirulence and antibiotic adjuvants in the past during my postdoc, and we had some success, and others too. But now the shift in my program is more taking a step back, understanding what's going on in terms of resistance and microbial virulence, and coming up with a pipeline of identifying more of these agents that target both resistance and virulence. And so before I talk about our how we approach this problem, I'll take a step back and talk about how traditionally antimicrobials are evaluated, and hence the antimicrobial discovery. And that's kind of, if we go through a patient with an infection and say severe infections, and how these are characterized in the clinical microbiology lab, you take a specimen from the patient and then go through the standard microbiology kind of approach, bacterial culture, isolation of individual pathogens that are kind of causing the infection, and then some identification, and then end up with an antimicrobial susceptibility testing. These are standard assays. One of them is MIC, minimal inhibitory concentration, or disk diffusion, whatever. At the end, there is a report that will be generated that says, well, this isolate from that patient is susceptible to these antibiotics. It's resistant to those antibiotics. So don't, like, that report goes back to the clinician, and then based on that, they will avoid certain antibiotics, and then hopefully can be able to prescribe some antibiotics. And none of that affects how the patient is treated. And because of that, a lot of the antibiotic discovery and research also are done for the most part for a long time under conditions that kind of follow the standard protocols for antimicrobial susceptibility testing, which kind of, if I sum up the kind of the main highlights here without getting into too much of a detail, is that if we look at MIC assays, that we test isolated bacteria, we test one microbe at a time. And then the second is that we use standard culture media, such as Mueller-Hinton broth, or MHB, which has this composition. Some might recognize it, but like the different media that are used, MHB or others, are kind of similar in that they're complex rich. Basically, they can grow microbes. So in this particular recipe here, for like MHB in particular, like this is beef extract, acetylrosate of casein, starch, water. And that's kind of, if you think about it, it's nutrient rich, can grow bacteria fine in the lab, and it's more something like that, which is like a happy meal of some sort, like a milk, the beef, the carbohydrates, and that sort of stuff. If you think about that, blend that, it doesn't really mimic the conditions that are happening in a patient in our own systems. So the lung, or urinary tract infection, or whatever might be the infection site. And also it's usually several organisms present, and they're surrounded by other chemicals that are produced by the host, or other microbes that could be co-infecting, or just the normal flora. So and understandably, and that's kind of there are several epidemiological studies now, that there are lack of correlation. So results from standard antimicrobial susceptibility testing often do not correlate with the clinical outcome. So in some cases, like cystic fibrosis patients, like lung infections of CF patients, there are sometimes is almost 50% lack of correlation. So it's almost as good as flipping a coin then to decide whether to treat with that antibiotic or not. So the standard assays are not really reflective for some of those reasons, and we see the result of that. And sometimes it's kind of the opposite. So you prescribe an antibiotic, it doesn't work, there is therapeutic failure. Sometimes, because that process that I showed in the previous slide, it takes some time. So sometimes in severe infections, they have to start an antibiotic therapy, the patients on antibiotics. And I hear reports from like the colleagues working in the hospital in Régina, that they sometimes also see the opposite, which like the reports of that too. They start an antibiotic, it's working, and then the report comes back say that it's not supposed to work. So it's the lack of correlation, which is great for the patient, but then what's going on exactly is not clear. So the hypothesis here, our hypothesis, is that there are determinants that are important during infection. They are only expressed during, like, in the patient, at the site of infection, but they're missed in that standard testing condition. And so we think we can expose and detect those determinants if we test under infection mimetic conditions. So we still need to do our testing in vitro. So we cannot jump head into like in vivo right away. We still need to do mechanistic studies and such. But we think that those determinants that are missed under standard conditions can be uncovered under infection mimetic conditions. So that's what we do. And to do that, we kind of look at the two kind of factors. There are media that kind of are more representative of the different infection sites. So we use artificial urine, stimulated wound fluid, CF artificial sputum, and human plasma or serum as examples of culture media that can closely mimic some of the conditions at the certain sites of infection. And then we also take into account the chemical landscape at the site of infection, whether host-derived chemicals or microbial-derived chemicals that are coming through interbacterial interactions, perhaps. And we look at those, the effects of these on microbial virulence and antibiotic resistance. So there are several chemicals we're interested in, such as polyamines, corticosteroids, heparin. They're present at the site of infection. Some of them are overproduced during infections. So that's kind of two broad themes in the lab. One direction is studying the infection mimetic conditions, looking at these culture media, and then the other is looking at the effects of certain chemicals of interest. And the interbacterial interaction is kind of somewhere in between there. And so in terms of virulence, also we're interested in biofilm formation at different sites. And we also use Galleria melonella, the larva infection model, because it's also amenable to high throughput in vivo, and also we have a skin model, burn model for it. And we grow them by the thousands in the lab, breed them, yes. And they don't require ethics, and well, they can, we use them for we can apply them in genomic screens and others. And we study, we, for the most part, focus on several of the priority pathogens that are causing the majority of infections, fatalities, and morbidities worldwide. Staph aureus, gram-positive, and then several gram-negative organisms like Klebsiella, Pseudomonas aeruginosa, and E. coli. And so what I'll do is that I'll talk about two vignettes representing each of the themes. And so the first is looking at the studying bacterial responses in the presence of the host chemical, which this one here is polyamines. So polyamines are produced by almost all life forms, with few exceptions. So we produce, need polyamines. They are involved in many important essential roles, but they are more importantly they're involved in the response, the innate immune response, and hence they're also overproduced during infection. But for any normal, for any producer of polyamines, including bacteria, polyamines are essential. So if you completely shut down production and import, say in a bacteria here, you get cell death. So there is certain level that is required for normal homeostasis, and then a little bit more that's they are usually involved in response to several stress conditions, including antibiotic response, and then too much, it's lethal. So in all producers, it's always there is a tight regulation of the amount of polyamines inside, regulation of the amount of production, import, and detoxification. And it's been shown that for bacteria, bacteria need to be able to resist high levels of polyamines to survive in the host. And there are several reports showing that bacterial response to and resistance to polyamines is contributing to increased microbial virulence. And so the objective here was to screen for inhibitors of polyamine resistance, or to prevent bacteria from detoxifying polyamines, because then in the absence of that, they become more susceptible to the less resistant, less virulent, and then less resistant to host innate immune response, and there are higher chances for clearance. So we thought that this could be a good target for antivirulence therapy. So perimolding, a PhD student in my lab started with a high throughput screen of basically synergy with spermine. So one of the natural polyamines. So basically we're looking for increased sensitivity to spermine. And she's screening the library of diverse small molecules, but also we had a subset within that, a bit of a focused approach, having polyamine analog library for more like rational-based approach for discovery here. So I'm pulling all together. From there, we had some primary hits. We characterized, like Perry did additional dose response, and then we found 6 synergists, and then finally we had those 3 prioritized hits. One was from the polyamine analog collection, 2 was from the diverse small molecule library. I'm gonna show just one example here of what we are seeing here. So that's a checkerboard assay where we look at the combined activity of 2 drugs here in general, 2 molecules, 2 antibiotics, 2 anything. And so dark blue here is growth, white is inhibition. And if you look at, start here, maybe the first column here shows the activity of one molecule alone. So that's the activity of spermine, and the lowest concentration, the minimum inhibitor concentration or the MIC of spermine for this. This is Staph aureus here, an MRSA strain, USA 300. The MIC is 20 millimolar. That's still a physiological level of polyamines that are in the millimolar range. And the x-axis here shows the first row is the activity of that compound, the screening hit alone. So an MIC of 20 micromolar. And then the wells or the squares in between shows the combined effect of the 2 drugs together. And what we are seeing here is that staircase effect, or there is a synergy. So in the presence of a small concentration of that compound, the MIC of spermine keeps dropping down. So it is the compound is inhibiting the ability of staph to deal with spermine and resist it. And so that's kind of what we're looking for. So that's, like I said, that compound is polyamine analog, and we did some small kind of small set of structure-activity relationship, and seems like the propylamine group here is important for the synergy. The longer that alkyl chain here, the inhibitory activity of the compound is. So we could see some trends here. But I want to point your attention that there are 2 kinds of activities that molecule is displaying. Growth inhibitory effects and synergy with spermine. So we wanted to understand the mechanism of the 2 activities. So starting with the effect of synergy, the polyamine synergy here. We started with a chimiogénomique screen of the library of Staph aureus, so screened the library of mutants. So this is a sequence-defined transposon mutant library in that MRSA strain. So each well that we tested, or each dot here, that's an individual mutant. So it's covering the non-essential genome of Staph aureus. And we tested that under in sub-inhibitory concentrations of spermine, so some of the resistance determinants of spermine, and we identified one known determinant, the one that is known to be involved in virulence before. One of the contributors to that polyamine related virulence was PG, which is an N-acetyltransferase, so acetylates spermine and spermidine, render both molecules non-toxic to the cells. And then we identified others also that has been not previously linked to the polyamine response. And so we tested the small molecules that the hits against those different polyamine resistance determinants. That's in one case, we're looking for lack of synergy. Those are mini checkerboards. I'm not going to get into the details of each of them, but that kind of shortlisted SPG as the potential target for that molecule. And so looking at the larger, the full checkerboard, so that's the wild type, we see that synergy that I described before. And then the mutant here, SPG mutant, as you'd expect, that it's more susceptible to spermine. So compared to the wild type of 20 millimolar, it drops down to around 2.5 millimolar susceptibility to spermine. But then now it's losing the synergy. So because the target for that synergy is no longer there is no longer synergy with the compound. So that again suggests that this molecule is targeting SPG. And we did the same in naturally expressing or not express, like SPG expressing or not expressing strains. So that's the USA 300 wild type, the CMRSA10 strain also carries the SPG. Those are by the way, Staph is a non-producer, so it doesn't produce polyamines. So the majority of Staph aureus do not carry that detoxification mechanism. The majority of Staph aureus do not carry SPG or cannot deal well with polyamines, and that's why they're less virulent. And so if we test that in those SPG lacking backgrounds, so they're more susceptible to spermine, but at the same time we don't see as much synergy. There's still a little bit of synergy, and not going to talk much about that because there's another kind of acetyltransferase in their backgrounds too. But for the most part, that suggested to us that that molecule is targeting that acetyltransferase, that particularly spermidine acetyltransferase. So to confirm, we did an enzymatic assay for those enzymes, and there are some controls here, heat inactivated controls. But then the molecule of interest, the main hit here, as we can see, dose-dependent inhibition of SSAT activity, or spermidine acetyltransferase activity. We tested the other two analogs that had one that had a partial synergy with spermine, and another that had no effect. And we can see that the one that had no effect in our whole cell assays also doesn't have an effect on SPG in vitro. The other one had an intermediate effect in whole cells, and also has an activity in against the enzyme. So again, that confirms to us that synergy, that polyamine synergy, is related to an SSAT inhibitory effects. So SPG is the target here. In terms of the growth inhibitory activity, we started that it must be targeting an essential target. So we started with a knockdown library of essential genes, but we didn't find a protein target initially. So that kind of ruled out a protein target, leading to that growth inhibitory effects. So that pointed us to membrane effects. So we did the DISC3 5 assay, and that showed that there is at those growth inhibitory effects, we see membrane effects too. So it's likely that at high concentration, the compounds, that particular inhibitor has a membrane effect, which leads to that growth inhibition eventually. And so we were then thinking that maybe if it's targeting membrane here, it will target also eukaryotic membranes, leads to cell lysis, which would become a problem. So we tested a hemolysis assay to see if there is membrane effects in eukaryotic cells. Seems like in the same concentration range, we're not seeing membrane effect in that hemolysis assay. At higher concentrations, we start seeing effects, but at higher concentrations, from the growth inhibitory concentrations, like the bacterial growth inhibitory concentrations, and definitely much higher than what is needed to synergize with host polyamines. And that is not near the signal we're getting with the positive control in that assay. Okay, so that was initially uncovered in Staph, and so SSAT and SPG is kind of conserved across bacteria producers and non-producers. And like I said, there has to be that tight balance, whether you're producing or not. So so we tested the inhibitor against first representative gram-positive and negative that we had SPG mutant in their backgrounds, and we can see that the same is happening. There's synergy in the wild types of Bacillus, E. coli, Salmonella typhimurium, Klebsiella pneumonia, and then the SPG mutant is susceptible, and there is no longer synergy. So we think that the same effects are happening in those other gram-positive and gram-negative bacteria too. So it's leading to that potentiation, and it's through SSAT inhibition. And so tested more bacteria that are anything that is producing and like carrying SPG homologue, like Enterococcus faecalis and faecium, Staph epidermidis, Burkholderia syncypacia, even Mycobacterium smegmatis. There is we see that synergy there. So that's kind of great. So there's potential for that broad spectrum activity as an antivirulence. And so the other thing about, so I mentioned in the first slide about polyamines that at sub-inhibitory concentrations of polyamines, they are good for different stress response, including antibiotic response. So we know that there is increased resistance of polyamine-mediated antibiotic resistance. So that we saw the same thing in Staph aureus, even though they're non-producers. The Staph aureus strains, so US A300 here, that is capable of detoxifying polyamines. It takes advantage of polyamines to increase resistance here. So that's kind of the opposite of the staircase that I showed before for the synergy. That's here what we call antagonism. So in the presence of spermine, Staph is losing, or vancomycin is losing its potency against Staph. So vancomycin is the first line treatment for MRSA bacteria. So now in the presence of spermine, there is that antagonism. But in a SPG deletion mutant, or in the presence of low dose of that compound, 2.5 micromolar, that antagonism is no longer there. So we see that square. So that's kind of that additive effect. There's that the inhibitor abolishes that polyamine-mediated antibiotic resistance. So again, that can suggest that it can work as an antibiotic adjuvant as well. So preventing the increased resistance. So we had a collaboration with Omar El-Halfawy from University of Alberta. They tested the molecule for us in macrophages. And so the SPG mutant had a defect in intracellular survival, and treating with the inhibitors, if you do pre-treatment or treat after infection, that led to eradication of salmonella infection, like the intracellular survival in macrophages. And Previously, I said, like I mentioned, it's known that response, like the polyamine response, is important for virulence. So it has been shown before in a mouse model that deletion of SPG leads to attenuation in this case, a mouse abscess infection, and that is related to the level of host polyamines. So that's driven. So if when they treated with eflornithine, which is an ornithine decarboxylase inhibitor, so reduces the production or biosynthesis of putrescine and then the rest of the polyamines by the host, that attenuation is no longer seen, because that's kind of coming from the host polyamines. And we're seeing the same thing in Galeria now. I have preliminary results that suggest that the inhibitor is actually synergizing with host polyamines, because when we reduce, we cannot completely shut down host polyamine biosynthesis because that would become lethal, but you reduce that host level of polyamines, it shows that that we're actually synergizing with the host, which leads to that of our kind of summary slash working hypothesis is that during, for example, a lung infection, typically there is kind of a higher level of inflammation, and that would lead to also increased levels of polyamines. And then if the bacteria are susceptible to polyamines and also you treat with the right antibiotic, there would be the antibiotic would clear the infection. But if there is that component of the chemical mediated effects that are going on, and then you have the bacteria that are resistant to host polyamines, and those polyamines would help increase resistance, like so lead to the polyamine mediated increased resistance, then you get a surprise in terms of the unexpected resistance, because like in a standard AST, again, like I said, you're not taking into account the host molecules, but then that leads to increased resistance to antibiotics and the host defense, so we lead to an unexpected therapeutic failure. So I think that if we combine with the PA resistance inhibitor, then you can kind of synergize with the host chemicals, so it can act as an antivirulence, potentially as a standalone, but most certainly it will prevent that polyamine mediated increased resistance. So again, will prevent that unexpected therapeutic failure of the antibiotics if you treat with an antibiotic. So it can work as antivirulence, antibiotic adjuvant. It's not always that polyamines increase resistance. There is some cases, in some examples, there is increased susceptibility too. So you have to know what's going on, and you can take advantage of that. And that's just as a quick example, some work from another student, Josh, has uncovered that the exogenous polyamines can synergize with some antibiotics, in this case, azithromycin, which is a macrolide antibiotic, typically used for gram-positive bacteria. So he saw the effect here against Klebsiella pneumonia, which is a gram-negative. And now he sees that there is, in the presence of polyamines, another one here, putrescine, another example, there is that synergy, which basically azithromycin is not supposed to work against Klebsiella, but in the presence of putrescine, there is susceptibility, so it can work. So he characterized the mode of action of that. There is a dual mechanism. I'm not going to get into the details for the interest of time here, but we took advantage of that to come up with so the understanding the mechanism led to potential targets that we can exploit, and he did that exactly, and found they did a small screen with the polyamine analogs and found some azithromycin adjuvants that synergize with polyamines at the low micromolar level. So we can really use that understanding instead of waiting for the host polyamines to synergize and do their trick. Now we kind of, because that can vary from one person to another, we can use that knowledge and in a more targeted way. So that's the first vignette. I'll touch upon a second one very quickly, studying the effects under infection mimetic conditions. And I'll picked one in wound infections. So, and the example I'm giving here for Staph aureus wound infections. Staph aureus is the most common bacteria isolated from chronic wounds, and chronic wounds are a huge. The 2% of Canadians are affected by chronic wounds. They cost healthcare. There's direct healthcare costs of around $4 billion annually, and so Staph aureus is the most common. There is also Pseudomonas aeruginosa, and we study that in the wound infection model as well. And so the hypothesis here that I mentioned before, so there are specific determinants that would be expressed in the host environment and not in the standard ones. So our goal here is to, so we use the simulated wound fluid that is more representative of the wound exudates. And the goal here is to uncover, well, first to understand the bacterial responses under these conditions, and then by knowing the different discrepancies between the results from the standard lab conditions, then we can uncover new antibiotic response determinants that are only observed under the infection mimetic conditions. So Colin Rieger, another graduate student, started by comparing, doing this antibiotic susceptibility testing in MHB and synthetic wound fluid, and he found in 55% of the cases there is discordance between the results. So he tested a whole bunch of antibiotics covering the different potential targets within the bacteria, and we call it when there is a fourfold or more shift in the MIC. That's kind of the threshold here. And so it happens both ways. So I'll just give one example here from what we've seen. In this case, doxycycline, a tetracycline antibiotic, a protein translation inhibitor. The shift happens. So in MHB, it's quite susceptible on 0.125 microgram per ml MIC. But then in the simulated wound fluid, the MIC jumps up to 4. So on 16, 32-fold shift, depending on the day. More importantly, it crosses the clinical breakpoint that you kind of decide whether that's the strain is susceptible or resistant. So if you look at the MHB result, that's pretty susceptible. If you look at the synthetic wound fluid results, no, that's resistant. So that's kind of the phenotypes that we're most interested about, those that would potentially lead to an altered therapeutic response. And that shift was not driven by an antibiotic sequestration in the culture media. So it's not kind of physicochemical. Okay, so he did a chimiogénomique screen, again, using that library. The examples that I'm saying today are from Staph, so the same library that I mentioned before. And from that, he was looking at the putative determinants of that increased resistance. And from there, he found, like, long story short, he found MNT-ABC as the determinant of that increased resistance. So one example here, what we're looking for here in a dose response is that, again, that's the wild type in the wound fluid and in MHB. And then the mutant here, that's MNTB, you see the shift back to the susceptible. So it's close as how MHB, the wild type is behaving in MHB. That same shift doesn't happen in MHB. So it's kind of that shift is only happening in synthetic wound fluid. So that's kind of specific. It's not a general antibiotic response happening in any media that we test. No, it's only happening here in the wound fluid. Same is happening in MNT-C and A to different extents. So it's that system, which can also gives reassurance of what we're seeing here. It's not a fluke or something. So that's MNT-ABC is a manganese transporter system. I'm not going to get into the details, but it's interesting because it is expressed in vivo. Like it's known to be, like Steph expresses it, it's overexpressed. There is also high antibody response because of that. And because of that, it was thought to be a good candidate for a vaccine. So it's somewhere in development in a 4-component vaccine developed by Pfizer. Some stage, I think it went as far as phase 2b/3. So it is relevant in vivo in a bacteremia model, in bacteria patients. Like that's the data from patients, and also in other in vivo settings. And so we know, like again, getting into the details, the kind of trying to know what's inducing that phenotype in the medium. We kind of know that this is a low molecular weight fraction from the synthetic wound fluid, from the fetal bovine serum, one of the components in the medium that mediates that doxycycline resistance. But so we thought that maybe it's manganese, something related to manganese that is driving that, and it wasn't manganese. So we did some sequestration, exogenous supplementation of manganese in different conditions, and it wasn't related to manganese. So we thought, well, maybe it's it somehow facilitates the doxycycline efflux from the cell in synthetic wound fluid, and so we compared the intracellular concentrations of doxycycline accumulation, and we see an accumulation of doxycycline in synthetic wound fluid in the mutant, but that's not happening in MHB. So it seems like it's playing a role here in the accumulation, leads to some sort of an efflux of some sort, or facilitating at least the efflux of doxycycline. We checked the phenotype against an array of other isolates, clinical isolates. So this is a panel of 10. I'm showing 8 here, but it's there are 10 Canadian MRSA panel of like the most circulating MRSA strains across Canada. The same phenotype is happening across the 10. One of them is also is a US A300 derivative, so we already know the answer to that. So, but anyways, and those MNT-ABC is conserved across all these CMRSA strains. So it's so the phenotype doxycycline is happening across all of them. The system is also conserved across all. So that's suggests it's a good potential target for an antibiotic adjuvant that would work in across different isolates, and also would prevent that unexpected increase of resistance. So there is a chemical screen that we're doing in synthetic wound fluid. I'm not gonna go into that, but I just want to highlight one project that we're interested in, and it is in collaboration with indigenous elders and knowledge keepers, where we collaborated with several from different communities across several treaty lands in different parts of the prairies. So this is Régina is right here, and that's Saskatchewan. And if you overlay that with the treaty lands, there's so we're in Treaty 4 land, but we have collaborations with spanning different geographical zones also. So different plant kind of like there is diversity of the specimen that we were collecting. So so that was an NFRF exploration project, and we screened those samples against different like planktonic and biofilm bacteria. So one example here is that we screen in the standard conditions, I guess in this case here, and also compare that to the infection mimetic condition. And in some cases, and that's true for the chemical screens too, we see activity across, right? In like in the two kinds of like the standard and the infection mimetic. But in some cases, the activity is only discovered under the infection mimetic conditions. So that's clearly why we're why we're doing this. We're missing, drug discovery is missing out when we're doing the discovery programs in the standard conditions. So it's just a couple of examples here. Nothing is happening in MHB, but there is inhibition in synthetic wound fluid. So that's just for the interest of time, I cannot go into that details, but the goal is to do also that's standalone antimicrobials, but we also go for like the combination to look for adjuvants and things like that. So in that part here, we start with a phenotype, look for those discrepancies, and then understand what's going on. So like an example here for that particular phenotype, we found MNT ABC, but we do the same for others. And that would give us putative targets that we can target for antibiotic adjuvants potentially, or maybe antivirulence agents, and we screen under infection mimetic conditions. So that's kind of the workflow, starting with the phenotype. That's from like CF sputum, and you look even like visually, it's quite striking the difference, but in the composition also is different, obviously. And that's another phenotype in CF sputum, and you then follow up with genomic screens, you come up with a underlying mechanisms, but also potential drug targets, and know what is happening in terms of the host effects, so that you can, we're interested in studying that too, so that we know what is kind of triggering that phenotype, and then drug discovery. And clearly don't do that by hand, like robotics, and there's some examples for what we do and use, but bringing it back to where I started, clearly the standard assays are not representative of the conditions at the infection sites. And so what we hope is to substitute, is to bring those other elements, studying infection mimetic conditions and the chemicals, the effects of the chemicals at the site of infection on the different phenotypes, the resistance and virulence. And hopefully that will lead to uncover clinically relevant druggable targets that we can, based on that, discover new antimicrobials, including antivirulence and antibiotic adjuvants. And so with that, I have now, we exponentially expanded in the, after a slow start initially during the pandemic, but then we have a larger group I'm proud to work with, really great and fun to work with in the lab, collaborators, and also the different funding agencies that are supporting my group currently. So with that, thank you very much again for the invitation and for listening to me, and I'll be happy to take any questions.
Hello? Yes,
yeah.
Great. I was wondering, when you did the discordance assay between MHB and SWF, did you see differences depending on the MOA? So where you didn't get shifts for typical, for types of MOAs versus others where you typically got shifts?
Yeah, no, that's a very interesting question, and we were initially, I was like looking for something like that because it will make it easier in terms of like what's going on mechanistically, but not so much. So obviously the drugs within the same class, we see the same phenotype, but then we cannot, for the most part, so far we cannot have a correlation of like that's peptidoglycan. So for example, sometimes we see a huge shift with beta-lactams, unexpected shift in MRSA, which is super susceptible in some of the media. But then vancomycin, nothing. But also it's not a perfect, there are differences because vancomycin works in an MRSA anyways, and vice versa. So we're not able to do that correlation. Same thing for protein translation inhibitors. We don't see the same shifts all the time, but yeah, interesting.
So in the
So I want to wrap my head around the polyamine story because within the gut microbiome, the bacteria themselves, part of their arsenal for microbial interaction are polyamines.
Yes.
So if you go in there and then say that, okay, I will add or subtract polyamines, how much of what you are adding or subtracting is relative to the effect of how much they use between themselves during their natural habitat.
Yeah, that's yeah, excellent point. I didn't delve into that. My initial interest in polyamines started in an interbacterial interaction. So actually, at that time I was studying Burkholderia synesthesia, and they interact with Pseudomonas aeruginosa in the context of CF. Both are cystic fibrosis pathogens, and Burkholderia would produce, like, both are polyamine producers, but because Burkholderia is more resistant, they are able to use that sub-inhibitory to them, like sub-inhibitory concentrations of the antibiotic that's super inhibitory to Pseudomonas, but sub-inhibitory to Burkholderia, overproduce polyamines. They protect themselves and other bacteria in their surrounding. So I guess it's context dependent. There are lots of variables there, but I would say if you're disrupting the detoxification or the production of polyamines, specifically to bacteria. So if it's a specific inhibitor bacteria, not to everything polyamine, because then it becomes toxic. If you disrupt it, then it's good for as a strategy, because then you're disrupting that balance no matter what. So they're using that to respond to antibiotics. So a low, like I said, in that kind of small strip of like that, too little, that's bad. Somewhere in between, that's good. A little bit more, it's good for bacteria. So they rely on that as an antibiotic resistance mechanism, and we're interested in studying that as well. So if you hit that balance, either by disrupting the detoxification with the example, like, it didn't matter if they're producer or not producers. It still threw off the bacteria in terms of the response to polyamines, but also antimicrobial response. Or if you inhibit biosynthesis for producers, it's still good strategy. The question would be then, in the context of the microbiota, are you worried about the microbiota, like so that you don't want to create that dysbiosis potentially? That's one consideration to think about. But in general, yeah, you're absolutely right. It's part of their strategy to resist, and then you can kind of. But any imbalance would be harmful for them, because the tight balance is important as they upregulate or down. Some.
Yeah, my question is a bit related to what you said. So about this synthetic fluid, is this something like a property of that fluid that is responsible for this? Or if you look at gene expression patterns, do you see a typical like virulence response that could, like that you can find a common denominator here in some way of different wound environments or different infection environments? Or is this super specific to this one environment and one medium, and we will never find out what.
Yeah, no, that's an excellent point. So I don't know right now for sure, but the goal is, so those media for like the wound or others, there are usually multiple recipes related, but with some variations. So we test across multiple. So we did that already in other, like in the CF sputum, there are different recipes out there. So we use that as a proxy, or like, okay, each one is independently developed to mimic the certain ingredient, like certain composition from a patient group. So by looking into those different compositions, so that potentially would look at like what might be happening out like in different patient groups, but that doesn't address that directly. Where yes, in some cases, like in the MNT ABC, one thing that we could look at is the kind of the expression levels or the data coming from patients, or expressions in vivo experiments, like that kind of look at what is going as far as like virulence determinants in real in vivo environment.
Aren't there some virulence factors so well known or some regulators that if you look at the regulators, see that these guys?
There are, and no, yeah, absolutely. But then, so I didn't directly mention that here, but so some would be like virulence determinants known from like under any environment, so including MHB or anything else that we tested. So these would be potentially known from before or uncoverable in the standard conditions or multiple other, not specific to that to the wound environment. So we screen also in the standard condition, but to kind of look at what is overlapping and what's not. I'm not sure if like that kind of virulence determines that the known virulence determines that what you're referring to. That's if it is universal, we probably would filter it out. Doesn't mean that it's not involved in whatever is happening in a wound, but it's not involved in the difference in the phenotypes. But I think the other part that I think you're getting at initially is the synthetic wound fluid is actually representative of the patients, and so we try to do so. The next step here is we test in the wound model here. We'll move that to an ex vivo skin infection model, so human skin. We're having that before that though. We had the Galeria burn model, which has been shown to kind of like correlate well with in vivo burn and wound models. So we try to take it one like from that synthetic medium to something in vivo, and then the ex vivo human skin and so on. So we try to do that in other models as well.
Salah? Your microphone. Salah, microphone.
Interesting talk, thank you. Maybe the people touch the models. I mean, as you stated, as you mentioned, 50% or more of antimicrobial results do not match with patients. One would think about the microbiota. It's well known that there's a family, there's microbes that are good and enhance the community literature. They enhance the antimicrobial effect. So, and there is metabolomes. There is some compounds in serum that that could be looked in the literature. I can't recall, but there is some compounds that we know if this tested biochemically in serum, if it's high or low, it means the good bacteria, the good microbiome is high or low. It's a notion. If can these compounds are reviewed and looked and try to produce it in the ex vivo model so that it match more in similar context. What I'm saying, CF, for example, cystic fibrosis patients are known to have problems in absorption of fat. They have nutritional issue beside the microbiota. So I mean, if these metabolomes of microbiota can be followed, where in the literature reviewed, where the ones that what compounds are known to distill the good microbiota that enhance the antimicrobial effect on the viral effect, and this can be, for example, simulated in the ex vivo. This you could bridge the gap onto the loop between the vivo and yeah. Also, for CF, if there's any nutritional issues that can be similar when you test for Pseudomonas aeruginosa that could simulate cystic fibrosis patients. There is a lot of work on diabetes featured in cystic fibrosis, these things. Sometimes the mutation is, I don't mean to go vivo, but it's at least it can have some feature that will can bring these correlations. Of course, you cannot do for any patients microbiota, but that you can go to
Yeah.
That's what because I think from what you said.
Yeah, no, thank you for this, and absolutely, all right. But I mean, we have to start somewhere, and we pick some, but there's definitely a lot of chemicals that are out there. Like we just they're yeah, and some of them are derived from microbiota, and others are host derived, and they are absolutely right. There are many of them, and sometimes they're specific to a certain condition. So and so we try to even for the interbacterial interaction, we try to do it in a simplistic way as well, like 2 microbes at a time, because that will enable us to study it mechanistically too, in terms of the pathogens that we're studying. So to get a sense of what is the influence of that interaction on the microbe side.
What's called combined effect.
Yes.
At some population, some conditions, there's combined effect of this antibiotic with some chemicals that could induce what is deficient in microbiota, good microbiota, that one or whatever. And so that it's have these conditions represented, and then this for in the future long term, if this patient has problem, they can look at as what in the serum for this test, and then see if we can take the antibiotic with this drug or with this or this. That's the long term. So
yes, you're suggesting something like if I'm sort of a supplement or something, like if there is a deficiency in one particular, yeah, or a metabolite that leads to, then we can combine that metabolite with the, absolutely, yeah, no, that's excellent suggestion.
Yeah. 2 questions. The first one is about the Gallinaria model, which is, or who don't know, an insect larvae.
Yes.
What do you want to do with it, and why this model?
So we actually do a couple of things with it. So first, it's good for, well, it's used for systemic infections. So we inject, and it's known to have an innate immune response and some cellular responses similar to ours. And so it's for in vivo infection, there is a good correlation between what you see in Galeria versus what you see in a mouse or rat. Not a perfect correlation, of course, because but it depends on what you're studying. But it's at least from the immune response, there are a lot of similarity with our immune response. So it's as an infection model, it has been used for systemic infections. So you inject, and then like the infection, with a dose of a particular inoculum of the organism, and then you track either survival, or you can also we can open it up and see the recovered CFUs from the hemolymph into the serum of these worms. So systemic infection, right? That's the most, and we can treat also. We can, after the infection, you can inject later on again with an antibiotic or something. And again, it seemed to work as a good filter before when we moved from Galeria, which is it's an insect that goes into a larva. So it's the wax moth, or like you can find it in a pet store. So the experiments that were done in Galeria, and then later on in vivo in an animal, for the most part they kind of matched. But then the second application is the burn, and more recently the cuticle of the worm seemed comparable, not comparable, but more representative of like a wound than a polystyrene plate, right? So you have that kind of exudates from, again, the hemolymph after you burn. So we're now optimized for a higher throughput burn infection model.
So that the resistance in Galeria is different.
We didn't look for evolution of resistance in the world. So it's usually the experiment. So the worms would last maximum 5 days or something, which I get. Yeah, well, you can study evolution of resistance. We didn't do it. I don't know if anyone did it, but it's interesting. So we, yeah. Yes. Yeah, exactly. And then if you're treating with something, you see if it's working or not in that kind of in vivo model. And it's good to compare between the different wild type and mutant or some sort if you're interested in a particular mechanism in terms of response to a certain antibiotic or of some sort. So I don't know. I mean, it would be very interesting to look at the development of resistance in a Galeria background. I don't know if anyone ever done that, but that would be very cool to do, to kind of understand, because there is still antimicrobial peptides that are produced and increased, like overproduced as any immune response would doing a certain infection. So we haven't looked, and I don't know if it's usually like an infection model you hit with the infectious dose or with a drug and then track the survival, and then because once they're dead, you cannot, well, they melanize, they die, and they cannot recover hemolymph from there any longer. So I don't know. You have to, even for the CFU counts that we do, we have to stop at a very early stage so that like 3 hours or something, where the first fatality, you'll see it a day after, depending on the dose, obviously, the inoculum. But generally, you look for CFUs from the hemolymph early on when the larvae are quite healthy at the time. So, but be very interesting to look at that in terms of, you know, like evolution-wise.
One last short question.
Yeah, very interesting work. I wonder if you can go one step back. Instead of testing the antibiotics in vivo or ex vivo, if you can add some immune cells in your media. I don't know if your media you can grow cells in your media. For example, you mentioned in that presentation that you were collaborating with someone who's using raw macrophages. Is that a possibility that you grow macrophages in the presence in one of those mimetic medias and look at by that you can add one more dimension, like inflammatory conditions, and see if this can make a difference.
Well, that would be very cool. We didn't do that. I don't know if how well they will grow in some of these media that we're using, but we do some. We done a screen in RPMI too. So as again another like a proxy, so that like cells can grow there, and we're interested also to study at some point survival in macrophages, and other people in different organisms looked into that, but we're trying to, well, at some point that's where we're interested to expand also for some of the pathogens too, to see determinants for intracellular survival in some of the organisms. So not exactly in these media, but in general, like the survival in macrophages. That's of interest. But, yeah, thank you.
OK, je vais remercier Omar El-Halfawy, qui parle très bien français d'ailleurs, peut-être pas suffisamment pour faire son séminaire en français. Donc, je le remercie, je lui souhaite bonne chance dans ses travaux. Je serai terminé en me joignant au doyen pour remercier M. Parizeau pour la vision dont vous faites preuve, je pense que d'investir dans la faculté, notre mission de développement, c'est vraiment extraordinaire. Je suis convaincu que la série de séminaires Télesphore-Parizeau va être de nature à favoriser les collaborations et puis dans les années futures, je pense qu'on va avoir de belles conférences. Donc, merci à vous pour votre aide. Merci à ceux en Zoom aussi d'être venus. Merci à tous. Au revoir.